Hey everyone, welcome back to Winter Seed Studies. I hope you all are doing well. So, we were discussing about cell biology, in which we have recently started discussions about cell signaling. So, we are going to cover the cell signaling part in very detailed manner. So, there will be a few lectures about cell signaling basics. So, yesterday we talked about the basic stuff like how what is cell signaling, how cells are able to communicate with each other, and what is the basic, what is the basic pathway that is followed for a cell to signal some other cell, right? So this is the schematic that we discussed yesterday in great detail. So we discussed that whenever extracellular signal molecule or any chemical signal is being released by some other cell, or it is present in the extracellular matrix, it goes and binds to its specific receptor protein, which could be present on either plasma membrane of the cell, which is also referred as cell surface receptors, or it could be present inside the cell, called as intracellular receptors. So those receptor proteins, once they bind to the extracellular molecule, means once the ligand receptor interaction is done in the cell, that activates certain intracellular signaling cascade. Okay, so that activates certain molecules that are present intracellularly, and which results into the formation of effector proteins. So we discussed about this yesterday in detail, like how what are the different aspects of this signaling pathway. So today we are going to discuss about. So we discussed about how a cell receives the primary message, the extracellular signal molecule, and it triggers certain intracellular pathway. Today we will be discussing about the intracellular signaling cascade in detail, the molecular switches that are present in cell, and how the cell, it is it is passing on the message that is being received on the receptor protein, right? So this is the receptor molecules, which are present on the cell surface. We are just taking example of cell surface receptors. So they are physically interacting with the ligand molecule, right? So once the physical interaction is done, they could bind covalently, non-covalently with the receptor protein, and that is going to activate receptor protein. So this receptor, what does activation of receptor protein exactly mean here? It simply means that whenever the ligand binds to any receptor, just like when the substrate binds to an enzyme, the conformational changes are seen in the enzyme. In the same way, receptor protein also changes its conformation. And once this conformation is changed, it could either undergo dimerization. Okay, it can undergo dimerization with same receptor protein, like same other molecule of the same receptor, or it could undergo phosphorylation. So it could get autophosphorylated, or it could trigger the phosphorylation of certain other molecule, right? So let's say that this receptor protein phosphorylates some enzyme, which is going to convert ATP into CAMP. Let's say that it triggers this reaction. So this, whenever conformational changes are done in the receptor protein, it is going to trigger some reaction, which is going to produce some intracellular signaling molecules, which are kind of the postman, which are going to take the letter out of the box, and they are going to take that letter to the post office, right? So they are like the middleman, which are going to relay the signal from the receptor protein towards taking it to the output proteins, right? Towards giving an output by the cell, forming the cellular products. So we are going to discuss about this intracellular signaling cascade that happens in the cell once the receptor protein gets activated. So what does we mean by activation of receptor protein? I already told you that the activation of receptor proteins means that either it could get dimerized, okay, either it could form dimer, or it could phosphorylate, it could get phosphorylated, means it could undergo autophosphorylation, or it could phosphorylate some other protein. Okay. Any of these outcomes are considered as activation of receptor molecule. So once this is done, it is going to trigger some intracellular pathway. So let's assume that this receptor protein, it has intrinsic enzymatic activity. So this this is a protein, right? So it is going to have extracellular domain, a transmembrane domain, and intracellular domain, right? So let's assume that the intracellular domain of this receptor protein is the one which has catalytic activity. So okay, so the functions of those domains are: extracellular domain is always for receiving the signal, the extracellular signal. The transmembrane domain is, for it is giving the structural integrity. It is holding that receptor protein in the plasma membrane, and that is why it is known as transmembrane domain. While the intracellular domain, why does it exist? Right? It exists because whatever signal is being received outside, that need to be relayed inside, right? And that is why this receptor has two arms, the extracellular arm as well as intracellular arms. It's like you are receiving information from one side and you have to pass it down to the other side, and that is why the intracellular domain exists. So this intracellular domain, it could form a dimer with the other receptor molecule. Okay, and once the dimer is formed, it might activate. So the whatever conformational changes are being done, they are being done in the receptor molecule, that could activate the catalytic domain which is present on the intracellular side, in the intracellular domain of the receptor protein, or the phosphorylation could activate the catalytic domain of the intracellular part of the receptor protein. Okay, or it could be associated with certain inactive enzyme, it could be coupled with certain inactive enzyme, and when it does the phosphorylation of that inactive enzyme could get activated, which can further produce some proteins. Okay, some proteins which are going to act as a middleman, which are going to act as the person receiving the later and passing that down to some other person, right? And those molecules that are present intracellularly, which are going to relay the signal, they are known as secondary messengers. Okay, I hope what I discussed here is it is clear to you all. So let's just summarize what happens. Whenever extracellular signal molecule comes, it binds with the receptor. Once this ligand receptor interaction is done in the cell, it is going to do some conformational changes in the receptor protein. That conformational change could be any of those three. It could form dimer. It could just change some conformation, it could get phosphorylated, or it could phosphorylate some other protein molecule. Whatever is being done here, it is going to result into formation of secondary messengers, right? Or it could result into activating the molecular switches. There are only two results of activation of receptor protein: first, formation of secondary messenger, or activation of molecular switches. Okay, I'll come to the molecular switches later. Let's first just discuss about the secondary messenger part. And why does this happen? What does receptor have in it, which is which makes it capable of doing that? It could be the catalytic domain, the enzymatic domain which receptor protein itself has, or it could be the coupling of this receptor protein with certain enzyme. It could be either of those. Okay, so this makes the receptor protein able to pass down the information which is being received from the outside of the cell, right? I hope whatever I'm trying to convey is clear to you all. So when this happens, when this catalytic domain gets activated, it could clear certain substrate, or it could phosphorylate certain substrate. So it could be a kinase domain, or it could be coupled with an enzyme which is known as kinase. So we have discussed about kinases and phosphatases in epigenetics. So kinases are the enzymes which phosphorylate certain proteins, certain substrate, and while the phosphatases are the proteins, they are the enzymes which are going to remove the phosphate groups from the proteins or their substrate. Okay, so usually those that they are referred as the molecular switches. When this activity goes on, that is referred as the molecular switches. Or they could create some some secondary messengers, or let's say that activation of this receptor molecule. Let's say that this is not a cell membrane, this is the membrane of endoplasmic reticulum, and the certain receptor on the endoplasmic reticulum gets activated. So it could signal the calcium molecule to either to get into the cytosol. So it will do the efflux of this calcium molecule from the ER lumen into the cytoplasm. So the secondary messenger molecules that are going to relay the signals, they could be calcium molecules, they could be cyclic AMP, okay, which are soluble in the cytosol, or they could be the lipid soluble molecules such as diacylglycerol, okay, which are present, which are just present in the plasma membrane, and they are going to do their signaling job being present there. Okay, those are the different types of secondary messenger that could be present. So this secondary messenger CAMP, okay, the change in the level of those secondary messenger is going to directly act as a, that it is going to directly act as a signal for the further process. Like, okay, so how does secondary messenger act as a signal, CAMP and all, that we'll discuss when we are going to get into detail of the signaling cascade. But for now, what I want to convey to you all is that whenever receptor protein gets activated, okay, it could undergo any of those phenomena, and when it undergoes any of those phenomena, it could either it could either activate the secondary messenger or do the formation of secondary messenger, or it could activate molecular switches. Okay, so we discussed about how the secondary messengers, what are the secondary messengers and what are different types of secondary messengers. Now let's discuss about what are the molecular switches. Okay, so so let's take an example. Whenever you receive a signal, let's say that now it's the night time and you want to switch off the lights, but you usually forget. So your mom, every day she calls you out to tell you that you have to switch off the lights. So that is kind of signal which is being received by you. So that signal tells you that you have to switch off the lights. So the switchboard is the molecular switch, and the buttons present on there. Let's assume that there's a switchboard and there's a button, that there's a switch present on that switchboard. So you are going to apply mechanical pressure or force on that switch so that it gets on and off. And once it gets on and off, it is going to either switch on the light or switch off the light, right? So it is going to convert the light bulb state from active to inactive or inactive to active, right? And for that you need a switchboard. In the same way, for activation of certain genes, there are some molecular switches that exist in the cell cytoplasm. So those molecular switches, they could get activated or inactivated, and they could result into the on or off of gene, right? And that is why they are known as molecular switches, because they in the signaling pathway, the only role that they perform is getting activated or deactivated. Once it get activated, they could switch on or off certain gene, and once they get deactivated, they could switch on and off a certain other gene, right? So this is the role of the molecular switches. So there are multiple categories of molecular switches that exist in our cell. The two major types that are very important and which are found in almost all of the cells are the kinase switches. Okay, there's no such there's no such terminology exists in the biological world. I'm just making it up so that it would be easier to understand. And there are GTP binding proteins. Okay, so there are two types of molecular switches, the kinase switches and the GTP binding proteins. So, well, what could be molecular switches? Again, they are proteins. So there are certain proteins which behave like molecular switches. Why do we call them molecular switches? Because they can get on or off to activate or deactivate certain genes. So they are very important in the signaling cascade, because well, that is what signaling is, right? You have to switch on certain gene or switch off certain gene according to the condition. If let's say the cell have to thrive under the stress conditions, cell have to thrive under the higher temperature, so it has to maintain its membrane fluidity. So it need to switch the metabolic activities of its own metabolic activities so that it can change the membrane fluidity. And for doing that, it needs to change its gene expression levels of that certain gene. Let's say that, like, in the previous classes of cell biology, when we discussed about the plasma membrane fluidity, we discussed that whenever temperature increases, we add the cholesterol in it so that it acts as the membrane fluidity buffer to maintain the to maintain the plasma membrane fluidity. So let's assume that the cell is undergoing some stress. The stress is the high temperature, and it wants to maintain its fluidity. It wants to make the cell plasma membrane rigid because the plasma membrane can't be more fluid. But we can't change the environment, right? So what we can do, we can change the composition of plasma membrane. And for changing this composition of plasma membrane, you need to change the gene expression levels. And for doing that, you need to have some certain, switch to on and off the gene, right? And that those molecular switches could be modifications that are done on the gene itself, on the protein that we sorry, not protein, on the gene itself, that we discussed in epigenetics. Or it could be certain molecular switches, the activators or deactivators, that they controlling the transcription. So there are activators of transcription factors, there are transcription factor regulatory proteins, right, that we have been discussing throughout the epigenetic series. So those regulatory proteins, controlling those regulatory proteins, for that we need some molecular switches, right? So that is what we are discussing here. I hope that's clear to you all. So that is why molecular switches play a very important role in cell signaling. So the kinase, so they are basically proteins, right, which are going to switch on the gene or switch off the gene according to the condition, according to the environment, according to the need of the cell. So the kinase switches, there's no such name, right? I mean, this terminology does not exist. I'm just making it up for you guys. So kinase switches, by that I mean that they undergo kinase cascades. And what are kinase cascades, right? So for that, let's first know what is kinase. So kinase is an enzyme which is going to add the phosphate group on any protein. So what happens is that when this protein, okay, let's say this protein this could have serine residue, threonine residue, okay, or the tyrosine residue. So any, not THS. Let's see. So it could have any of the residues, serine, threonine, or tyrosine, right? So any of this residue, all of them, what is the characteristic that they have? They have a free hydroxyl group over which a phosphate group could be added, right? So the proteins which have those amino acid residues, on those proteins kinases can work so that they could add phosphate group on the OH. Well, we already know about these exception to this rule, that in the sodium ATPase pump the aspartate residue could get phosphorylated. Well, that's a different thing. Here I wanted to tell you that those kinase switches, they are the proteins which has those residues. So they are, the kinases, they are categorized into two different parts, right? The serine threonine kinases or the tyrosine kinases. Because some kinases can work only on serine threonine, while there is other category of tyrosine kinases, right? So those serine threonine kinases, what they do, they phosphorylate serine or threonine residue present on the protein. So let's just draw what happens. So this is a protein, okay, and let's say that this has any of those residues, either serine. Let's say that it has serine residue. So on the OH, on the free OH of serine, the kinase works, right? The kinase works and it phosphorylates the protein. So it is phosphorylating the protein. So I'll just write pH. It is phosphorylating the protein. And once this phosphorylation is done, so whenever you add any chemical moiety on any protein, we discussed this up in the post-translational modifications, right? Whenever you add any group, like phosphate group, acetyl group, methyl group on any protein, so addition of any chemical group is going to result into the conformational changes of protein, and that conformational change is going to result into the difference in the its own chemical activity, right? So it's, for example, like if your appearance changes, right, it is going to change your confidence as well. Most of the times, let's say that a certain day you're just, you're just laying down and you're just being, you are just being lazy. So that is going to change the confidence that you have, right? No, that's not a perfect analogy to tell you. let's just assume that whatever physical, for physical changes are being done in the protein that are going to affect its chemical change. Like for example, whatever appearance you have, let's say like for some day in the lecture you are wearing some normal casual clothes, right? And that day you will you are just you are just doing your regular day-to-day routine in your college day, in your lectures and all, but certain day you have a presentation, so you dress formally for that. You have, because you are going on a stage and you have to perform, right? So why do we do that? Whenever you you wear formal wear, so that changes your appearance, and that brings out, that brings more confidence in you, that enhances your personality, and that enhance boosts your confidence basically, and that makes you look presentable, right? And that is why in the formal settings you often wear formal attire, right? So whatever physical changes you're doing to yourself, that is going to result into your actions, in your way of in your behavior, in your nature, the way you are behaving. You will behave casually the day you are wearing casual outfits in the college, but the day you have presentation, you are wearing formal outfits and you are behaving more formally. I know that this example does not sound perfect, but right now I can't recall any other analogy. What I want to convey is that whenever there's some change in the physical, in the conformation of the protein, whenever it changes physically, the physical interaction, the covalent, non-covalent, van der Waal interaction that the protein has with the in between the amino acid residues, that changes. Once it is changed, whenever a chemical moiety is being added on the protein, those physical interactions that protein has with the amino acid residues, that changes, right? Means it is changing its physical conformation, and when the physical conformation is changing, the function that is going to do that also changes, right? Because that function is governed due to the physical characteristic it has. What is an enzyme? Enzyme is a protein, right? So that has, if you change the conformation of an enzyme, it cannot do its catalytic activity, because maybe whatever moiety you have been adding, that moiety hinders the active site. So now on the active site, the substrate can't bind, and that is why the enzymatic activity disrupts, right? So whatever changes physically are being done in the protein, that are going to mirror itself into the chemical function of that protein. And that is why whenever a phosphate group is being added by the kinase, by this enzyme, that is going to change the state of this protein. So let's say that when it was dephosphorylated, so this is dephosphorylated state and this is phosphorylated state. So it could whenever it was dephosphorylated, we call it as it was inactive. That protein was inactive. But now that protein is activated due to the phosphorylation, right? It is activated. So once this protein is phosphorylated, it can either, activate certain enzyme. It can directly activate certain enzyme, or it could do the phosphorylation of other proteins, right? It can opt for, there are two choices for this phosphorylated protein, for this activated protein, that it could activate enzyme after that, or it could, continue a downstream cascade where it is going to phosphorylate other proteins. So it's like, you remember in chemistry, a formation of a free radical, it just promotes formation of other free radicals, right? When a certain free radical is formed, let's say that oxygen free radical is forming in a system, so it is going to take the electrons from the other oxygen as well, and it is going to, promote the formation of free radicals. So it acts like a trigger point. Formation of one free radical acts as a trigger point and results into formation of multiple free radicals. In the same way, the formation of, the formation of phosphorylation, the phosphorylation of certain protein, it could trigger the for phosphorylation of other proteins. And this cascade, this step-by-step process, let's say that this is protein, it gets phosphorylated, so it is going to phosphorylate certain other protein. So once this other protein is phosphorylated, it is going to phosphorylate other protein. So this whole pathway is known as kinase cascade. Okay, because it is not random. It is a series of step by step process. So a cell is following a repertoire. It is not doing randomly, doing things randomly. It is not doing the choices randomly. It is following a repertoire, and that is why this is known as kinase cascade, because the kinases are working on the proteins to phosphorylate them. Okay, so what happens eventually, that let's say that this goes on, one protein phosphorylates other phosphorylates the another one. So this can either now activate some certain enzyme, right? Or it could again phosphorylate certain other protein. But eventually it is going to, what is the outcome? It is going to phosphorylate certain enzyme, or okay, which is going to bring out change in the metabolic activity directly. Or what could it do? This protein itself could be an activator for certain transcription factor, or it could be inhibitor of certain transcription factor. Okay, or it could activate, or it could again, sorry, not activate, it could again yeah, well, phosphorylation means activation, or it could phosphorylate other protein, right? So eventually, like after following the series of this cascade, eventually there will come a point where the phosphorylation is going to result into the enzyme activation, or it is going to result into the activation of activator or activation of inhibitor. Once the activator is activated, it is going to, you know, activate the transcription factor, which is going to, this is a gene which is going to do the transcription of certain gene. Or if the inhibitor is being activated by phosphorylation, so it is going to inhibit the transcription factor, which is going to result into the, inhibition of gene transcription, right? So these are the outcomes that could come out of the kinase cascade. That is how the phosphorylation acts as a switch on and off for certain gene. So whenever phosphorylation is done on a protein, we say that it is getting activated. Okay, so I hope how the kinase switches work in the cell, that is clear to you all. Let's now discuss the other category of molecular switches, the GTP binding protein. And this is very crucial, because when we are going to discuss about GPCRs, this point will come again. So please focus on what I'm going to discuss in GTP binding proteins, right? So the way, phosphorylation is very important. It is an important event for cellular signaling, because it is going to act as an activation for certain protein, right? So what are GTP binding proteins? So it's very obvious from the name that those are the proteins which are going to bind GTP, right? So what happens is that, let's say that this is a protein, okay, and to which GTP is bound. Okay, let's assume that GDP is bound to this protein. So the GDP bound form of the protein is known as inactive form. Okay, it is inactive because it is not capable of doing any catalytic activity, or it is not capable of triggering phosphorylation of any type of protein. That's why it is known as, it is determined, or it is referred as inactive state of the protein. So this is a protein. Okay, so this GTP, let's assume that this GDP is being replaced by GTP. Okay, mark my words, I'm not saying addition of phos. So you might know, right? GTP. It's a guanine triphosphate, right? Guanine nucleotide triphosphate. So it is a triphosphate, means it has three phosphate groups. It breaks down into GDP with an inorganic phosphate, right? So mark my words when I'm saying that GDP is being replaced by GTP. Okay, so we are not adding phosphate here to make it GTP, but we are directly removing this GDP. We are removing GDP. GDP is removed, and we are adding GTP instead. Okay, so we are exchanging the GDP. Right, so we are replacing the GDP with the GTP, and that is why the enzyme that works in this condition is known as GEF, the guanine nucleotide exchange factor. Why exchange? Because we are literally doing the exchange, right? We are exchanging the GDP with GTP. So that is why this enzyme is known as guanine nucleotide exchange factor, GEF. Okay, so once Jeff acts on this GDP bound state of protein, this protein, okay, so here this now is bound to the GTP. So this state of the protein, the GTP bound state of the protein is known as active form. So now the protein is activated. So this could, again activate certain enzyme which is going to bring out metabolic change, or again it could activate the phosphorylation cascade. So it can do multiple things, but right now I'm not going to discuss that. We will discuss that when we are going to discuss GPCR in detail, and we will discuss that in that lecture. So this is the way the active form goes to the, sorry, inactive GDP bound form goes to the activated GTP bound form, right? But this reaction is reversible. Okay, so this is reversible. So once, but now you have to switch off this protein. So you have to inactivate this protein. So for that, you have to activate, this protein, this GDP binding protein. This, it has, intrinsic activity known as GTPase activity. So as the name suggests, GTPase means this is an enzyme which is acting on GTP, and it is an enzyme which is going to do the lysis of GTP, right? So whenever there is ase as the suffix, means this means that it is an enzyme with this substrate. The prefix is usually the substrate. So this is an enzyme. GTPase is an enzyme which works on GTP and which hydrolyzes the GTP. Right. So now this GTP will be hydrolyzed. So one inorganic phosphate leaves, right, and this GTP will be hydrolyzed, and it will be, again it will be converted back to GDP. So let's now discuss about this protein, this GTP binding protein. What is the conformation of it? So what is that? This is a heterotrimeric protein. Okay, it has alpha subunit. Okay, it has a beta subunit and it has a gamma subunit. So what happens is that, and as I say, that this protein has intrinsic activity of GTPase. So the subunit that has GTPase catalytic domain is the alpha subunit. So this alpha subunit has GTPase activity. Okay, so this is the G protein. This is known as GTP binding protein or G protein, right? So this, to this G protein, so what I've drawn here, this protein is this. Okay, so it is a heterotrimeric protein having three subunits, alpha, beta, and gamma. Alpha subunit is the one which has GTPase activity. So what happens is that, let's discuss it again. So to this GTP binding, so this is G protein, means GTP binding protein. To this G protein, the GDP is bound. So this is an inactivated state, right? So once the guanine exchange factor, Jeff, exchanges the exchanges the GDP with GTP, it just replaces GDP with GTP. What happens is that it activates this G protein, and the activation means it activates the GTPase activity. So it's once the GTPase activity is activated, the alpha subunit is split, and beta and gamma subunit is split. So the G protein splits into alpha and beta and gamma subunit. So it breaks down into alpha subunit and beta and gamma subunit, where the alpha subunit, the GTP is bound. Okay, so this GTP bound alpha subunit is deemed as the activated subunit, and this is now going to trigger the downstream signaling cascade, which we are not going to discuss that here, which we will discuss in the GPCR lectures. So what I was trying to convey here was the G protein, it has intrinsic GTPase activity. The alpha subunit is one which has GTPase activity. So this alpha subunit, it is going to, it is going to need a protein known as, for hydrolyzing this GTP and being able to convey the downstream process. This alpha GTP bound alpha subunit needs a protein known as GAP. GAP is GTPase activating protein. Okay, so this enzyme, the GAP enzyme, when it works on this GTP bound alpha subunit, so it activates the GTPase activity of alpha subunit, right? So like here you can see when the GDP was bound to the GDP was bound to the to this G protein. At that point of time, alpha subunit had the GTPase activity. But for triggering this GTPase activity, you need gap, right? So GTP bound alpha subunit, when the alpha subunit binds to GTP, only then gap works on this alpha subunit. And what does gap do? Gap triggers the alpha subunit ka the GTPase activity of alpha subunit. So and that is why it is it is very evident from the name GTPase activating protein means gap that activates the alpha subunit GTPase activity. So now this GTPase is broken. So this GTPase is broken down. Okay, so inorganic phosphate leaves, GTP will be hydrolyzed by the GTPase activity of alpha subunit and it will get converted into GDP. So once it is done, so this is how the switch on and off works. So this was the inactive state and this is the active state, right? So this is like switching on the protein, the GTP protein, and this is like switching off the GTP protein, right? And switching on the GTP protein could switch on or off certain gene, right? It depends whether it's activating the activator of gene, whether it's activating the inhibitor of gene. So the result could be either switch on and off of certain gene, and that is why GTP